So, a warm welcome again to the Kiel Imaging Seminar. It's great to have you all here today. It's my pleasure to introduce our speaker today, Dr. Patrick Vogel. Dr. Vogel is a habilitated researcher at the Department of Experimental Physics 5 at the University of Würzburg and the CEO and co-founder of Phase Vision GmbH. For over 15 years, he has driven research and innovation in magnetic particle imaging (MPI), an emerging imaging technique that utilizes dynamic magnetic fields to visualize magnetic nanoparticles. His groundbreaking work led to the development of the first traveling wave MPI system, capable of real-time imaging within a human leg, and to the creation of compact and mobile instruments such as the MPS mouse, which inspired the mobile COVID rapid test compass. Building on this success, his current project, Stroke Cap, focuses on creating a portable system for early stroke diagnosis, paving the way for mobile and point-of-care medical diagnostics. Before we start, please wait with your questions until the discussion. And with this, Dr. Vogel, the stage is yours.
Right. Yeah, thank you so much. It's a pleasure to present my work. And you heard about it the last 15 years. I'm working in this field here in Würzburg at the university. And yeah, when I started 15 years ago, this field was so new that nobody knows what we can do with that. And so it evolves a little bit. And I want to take you on a journey to the last 15 years where when we started to build up the first preclinical MPI system. And a couple of years ago, we also have the chance to enlarge the systems to the first human-sized systems. And yeah, let's see how this was done. So from concept to clinic, and it was a long way, but it's also quite interesting way. And magnetic particle imaging, as the name said, we are using magnetic particles for imaging. And so it's now it will go. Okay. And what's so these nanoparticles, magnetic nanoparticles, you have a lot. There's a lot of applications you can use these nanoparticles, MNPs, and for example, for imaging, well known from the MRI community as a contrast agent, and also for MPI, where it is a tracer, or for therapy, so drug delivery, there's this possible, and hypothermia, it's really interesting. It's also quite near to the MPI field. So just using um, magnetic fields, sinusoidal magnetic fields for heating up nanoparticles, and also in vitro diagnostics, so antibody detection. And 5 years ago, when the pandemic starts, we also had the idea to try out how we can use this technology for providing a real rapid test. And how this compass system is working, I will provide you in a few slides. So maybe we start with a little bit more familiar background. So the MR side here, for example, 7 Tesla whole body system and magnetic nanoparticles were also be used in this field a couple of years ago. There was the ferrocarbodran or Resovist, and when the iron oxide nanoparticles are brought into an MR machine, then you get quite interesting effects. You get an contrast, a special contrast, or in other words, you get a decreased signal. And you can see that here. And this was the first idea we followed 15 years ago. Okay, so with MPI, which is a tracer-based direct imaging method for these nanoparticles, we can maybe distinguish between, okay, is there an MRI? Is there a signal loss because of some artifacts, or is there a signal loss because there is iron oxide nanoparticles nearby? And we built up the first hybrid MPI-MR machine just to show that and to demonstrate this. And it was my work with the traveling wave MPI system, and another PhD student at this time. He built up the low field MR system, and so we could then brought together both systems and generate these nice in vivo hybrid imaging of 3 delicious Bavarian grapes. And one of these is filled with Resolist. So you can see here the 3 grapes quite nicely. And with an overlaid MPI image, you can also distinguish the magnetic nanoparticles. And then, okay, these signal drop here on the MR side, it's definitely caused by the presence of magnetic nanoparticles. So that means on the one side, and this is quite important, MRI is not MPI. So a lot of people come, MPI, yeah, it's quite similar. It's similar, but the only similarity is we are also using magnetic fields. Yeah, so an MRI, of course, you know that you are using a constant homogeneous magnetic field for the B0 field, and in MPI we are using quite rapid moving gradients for the imaging process. And how does it work? We will come also in a few slides, but let's start with the magnetic nanoparticles, just to get an idea how the signal is generated, how the basic idea, the basic physics is done in the background. So all starts in 2025, so 20 years ago, with this publication, tomographic imaging using the nonlinear response of magnetic particles. And nonlinear response of magnetic particles, but the first is what are magnetic nanoparticles? And there are particles mainly from ferromagnetic materials, so iron, nickel, or cobalt. And when we are talking, for example, from superparamagnetic iron oxide nanoparticles, then we are talking about quite small nanoparticles. So these particles are in a range of 10 to 30 nanometers. And you can see that when you start with a huge bulk of material, so iron material, and this bulk consists of a lot of areas, domains where the magnetization is constant in one direction. And when you divide these large particles into smaller ones, and you can end up with a small particle which is exactly the same size or has the lower size of these magnetic domains, then you are in a superparamagnetic regime. And these tiny little magnets, these are quite interesting because you can use them for fancy stuff. So let's start with you put together a bunch of these nanoparticles, these tiny magnets, and without an external magnetic field, all the particles are aligned stochastically and equally distributed in all directions. So the magnetization of this ensemble is 0. But now, when you bring this bulk material, this ensemble in near magnetic field, then all the nanoparticles start to align the magnetic field, and the magnetization of these ensembles starts growing. And when all the particles are aligned, then the magnetization goes to a saturation magnetization state. So as a physicist, of course, we're happy to show that in a formula. And you can see this here in this wonderful nonlinear function, the so-called Langevin function. And this function is quite cool because small changes in the external magnetic fields causes really high changes in the magnetization of the ensemble. And how we can use this. So when you now bring this ensemble, this magnetic nanoparticle ensemble within a sinusoidal magnetic field with a specific frequency and which is strong enough to reach the saturation magnetization of the nanoparticles, then you can, over time, follow the magnetization of these nanoparticles. And what you can see that this magnetization over time now does not look anymore as a sinusoidal magnetic field, the magnetization. So it's more an rectangular shaped system. And we are also using a coil system for inductively measure the signal, which means we measure the derivative of the magnetization over the time. So what we can see, or what we should see, is this peak-like structure. And after Fourier transformation, this is really cool, you can see that you start with a specific frequency, and the signal we get from this material consists of higher harmonics, in this case, odd higher harmonics of the excitation frequency. And these higher harmonics consist of all the information of the particle system and are really, really specific, or in other words, you can see it as a fingerprint of the particles. And this is the signal we want to use. And here, for example, you can see it for three different particle systems, so perimag and ferraheme and magnetosomes. Magnetosomes are quite specific particles produced by magnetotactic bacteria. And I have a small picture of these. And all the particles looks a little bit different. So, for example, when you look over the time, so on the x-axis you have the time axis, you can see that the response, so the derivative of the magnetization over time shows a different position, or different time points, different responses. You can also have a look in the Fourier spectrum, and you can see it looks also different in the decay of the higher harmonics. And you also can produce some hysteresis loops. To be careful, that are dynamic hysteresis curves, so are not comparable with the static ones. But we are using all these visualization tools or mechanisms to get an idea about the particles and of the particle, or the specific functionalities of the particles. For example, of course, from the hysteresis loops, we can learn the particle performance for heat dissipation, used for hypothermia. We can use the spectrum for the MNP fingerprinting, and we can also use the time representation of the signal to get an idea about the spatial resolution. So, fun fact, the magnetospirillum, these are here the bacteria. These little black dots, or more cubes, these are the particles which are produced within this bacteria. And we have a collaboration with a group in Bayreuth, and they're producing these bacteria. And it was the micro of the year 2019. I never know that there's something for every year for microbes. But what is the reason why these particles look so different? There are two basic effects. One is the Neal relaxation effect. What does it mean is that the magnetization inside the particle can follow external magnetic field changes. And there's also the Brown relaxation effect. And the Brown relaxation means that the entire particle, the whole particle, can follow the external magnetic field. And it turns out that for for smaller particles, the Neal relaxation is the dominant part, and for larger particles, the Brown um, relaxation is the dominant part. And so in the range between 20 to 30 nanometers, that's the size where both relaxation effects have an influence on the particle response and magnetization response. And you can also see from the parameters which influences the relaxation times. There are different parameters from the particles which influences these times. So, for example, the size or the shape. Also, this is important. This is quite interesting. The viscosity of the environmental media, or also the temperature. But also the size distribution, the MNP core structure. So we have single core particles or also multi-core particles. We have chemical interactions, and we have also particle-particle interaction. So it's a really interesting field for physicists. So this particle behavior. But what I want to say is a lot of parameters can influence the signal, which can be quite annoying, but often you have to see that as a chance, because you can use these effects for different stuff. And one of them was in 2020, and was our compass idea. And I think you can remember these days. So we have a lot of different rapid tests and methods to get an idea if you are sick or not. And we also came up with the idea. So okay, let's do that again. Let's do that also with magnetic nanoparticles. And the idea is quite simple. You have a magnetic nanoparticle, and you put a coating, a coating for biocompatibility and stability around this nanoparticle. And you also put some target, which, for example, is specific for some bindings. Again, I don't know, antibodies or antigens, also on the surface of the particles. And when here the antibodies are nearby in the medium, then the antibodies can connect to the surface, and then something interesting happens. So you can see it here. We have a quite small animation. So on the left side, you have the standard particle, so just the coating. And on the right side, you have the particles with the attached antibodies. And now, when we put both in the same magnetic fields, then you can see that the larger one, the particle with the bound antibodies, has a different dynamic. So it follows a little bit differently to the external magnetic field. And it's quite similar with how it's called, these ice dancers. So when you put your arms far away, then you are slower in the rotation against when you have the arms nearby on your body. And with this effect, we were able to distinguish between is there the antibody for against COVID in these days, is this there in the medium or not? And it was amazing that we could show that with a really small portable system. And this is the real COVID test, or real rapid test we built up. Where we reach the sensitivity in the picomolar range. And the fascinating part here is this compass measurement is really fast. So within the 10 seconds, you get a simple, robust, and easy signal. So for comparison, ELISA tests requires about 18 hours to get the same result. But unfortunately, we took too long to understand this effect. So we finished at the end of 2023. And so the pandemic was over. So maybe there's another one on the horizon. So we are prepared now. So that means magnetic nanoparticles are quite different and provides a lot of interesting effects. And um, but it should be seen as a chance. Yeah, you can use these features for different stuff. Um, we can also simulate the signals, which is important to just to understand what is the influence of different parameters. But don't want to go to the details here. And as I mentioned, um, often it's not a bug; it's a feature. So you can also use these annoying different parameters for maybe some new applications. Um, Another question, of course, when we can use this for some spectroscopic things, then the question occurs: can we also use this for specific imaging of targeted um, maybe cells within living animals? And for that, of course, we need imaging. So the question is now how we can encode these three um, ensembles of magnetic nanoparticles within this small rodent. And when I started 2010 building or thinking about the first MPI scanner here in Würzburg, we came up with this idea. So we started with 16 electromagnets, so simple coils, and we put them into this arrangement. And now, when we apply a sinusoidal current to all of these coils with the same amplitude, with the same frequency and the same phase, then you can generate a magnetic field um, inside this array, which of course follows the excitation frequency. And according to the nonlinear magnetization curve from this red one, the Langevin function, the magnetic nanoparticles or ensembles will also follow the external magnetic fields and show the magnetization over time. And you can see it here. So when the magnetic field goes in one direction, then the magnetization in the ensembles also increases. And when the magnetic field goes to the zero, then the magnetization flips into the other direction until it reaches again the saturation magnetization. But so now adding another coil. So here a receive coil, and you can see here live the signal, and what we are looking at is the derivative of the magnetization over time. And you can see here, the flipping of the magnetization process when the magnetization of the ensembles changes. So congratulations, this is not an imaging system. So we now build up spectrometer. So we can just see is there some particles inside or not, but it's not imaging so far. And to do that, we now change the current distribution over the gradient array. So we call this the linear gradient array. And we change the current distribution in a way that we can generate a magnetic field which follows this sinusoidal shape over the field of view. And you can see it now in this picture. So all three ensembles now show different states. And by applying now again a frequency, sinusoidal, to produce a sinusoidal current, but now with different phases between the adjacent coils, then you can produce a traveling wave, a traveling magnetic field. This is why the system is called so, obviously. And you can see over time the magnetization of the ensembles flips. Yeah. So these these zero crossing areas, here with this plus and minus symbol indicated, are called the field-free points. And when a field-free point now is traveling or is running through an ensemble, then the magnetization against, according to the Langevin function, flips from the one side to another side. And this flipping process produces again a peak in the receive coil, and now we can encode along the symmetry axis within an MPI scanner these three ensembles. So of course we want to do that also in 3D. So we then let's open up the scanner. So you can see here again the 60 coils, which are generating this traveling magnetic field through the along the symmetry axis. And we then add a saddle coil system, one for the X direction, one saddle coil, a second saddle coil system providing a field for the Y direction, and of course our receive coil, and we put everything together. And with this system, with this coil arrangement, we are able to move the field-free points generated by the blue coils, so by the gradient. We can then displace them in all the direction and generate a 3D scan of the system. So we built up the first system, and it took us about 3 years to build up the first MPI scanner, the first TW MPI scanner here in Würzburg. You can see it on the right side. There's the core system, and you can see now not only 60 coils, you have 20 of these coils, and you need additional 2 coils at the edges, at the ends for decoupling the system. And here also we're using 3D printing more than 10 years ago to generate these quite complicated saddle coil systems, just to make sure that everything is small enough to fit into the bore of our system. I found a movie which is really, really old, about 12 or 13 years. And it was one of the first days where we are able to run the traveling this dynamic gradient system. And it was operated at 60 amps. So it takes about 5 kilowatts of power and produces 80 millitesla magnetic fields, and has a traveling speed of 1 kilohertz. And just have a look. You can see here the system. So it's put in an aquarium just for cooling. But there's no fluid inside. We put in a wrench, and then you can hear a quite nice sound. So, and this was the sequence running or producing this traveling magnetic fields. And so this traveling magnetic field also produce some kind of forces, and you can also use this kind of scanners for other stuff, which was also quite interesting to see. Then of course you need some more equipment. So for the to run the system, of course, some control PCs and function generators. For the transmit chain, you need some amplifiers. We are using normally in the MPI community common audio amplifiers, because the frequencies we are using are in the range between 1 kilohertz up to 25 kilohertz. And then with some high pass and low pass filters and so on, just for producing the sinusoidal or clean sinusoidal excitation currents to produce the magnetic fields we want to use, we want to have. And on the other side, a receive chain with of course receive core, some additional filtering systems, and an ADC. Here, it's a digital oscilloscope these days, and this is a quite early view on the TWMPI scanner. So here's the scanner, of course, with a copper shielding. Here's the PC for controlling the sequencers. Here you can see some audio amplifiers and then our oscilloscope. And with that, we were able to produce the first images. I mentioned this a little bit, so you can see animation for 2D for covering a 2D slide. So these dark areas here are the field-free points, and by using on the one side you have the traveling frequency from the left to the right. So here's the gradient system. The red coils are one of the subtle coils, and the subtle coils is then used to move or displace the field-free points on a sinusoidal trajectory, and then you can cover a slice, one slice out of your scanner. Can also put or use the second saddle coil just to slightly rotate the trajectory, and also can acquire 3D images within the same system. So quite early pictures out of the system, and you can put in everything you can find. And we also did this here, for example, also a small rock I found during my holidays, and also put this into the scanner, and we found out that there is something inside which looks quite interesting, and this is how we started to use these systems. So this brings me to my summary too for the hardware. So the traveling wave concept, as I mentioned, you're using dynamic linear gradient array for generating a traveling magnetic fields. Everything was handcrafted at these days, and we are using just common audio amps, which are quite inexpensive on the one side, but also effective for running such systems. And then, of course, we have to calibrate, or have to use these systems. And here, for example, the sensitivity and the system. So MPI on the one side is a really, really fast imaging technology. So only a few, about 20, 30 milliseconds you need for a whole body scan. And from this side, we also want to optimize the sensitivity. We can reach quite nice, quite low sensitivity for magnetic particles. It's quite rapid, as I mentioned, this technology here, for example, this is a real time. It's a small bolus which is shooted through the scanner, and you can see it's also possible to reconstruct the data directly, and you can visualize everything almost in real time. And we also can produce high spatial resolution. Here, for example, we reach with a specific system about 330 micrometers. Nanometers would be good. Micrometers, and it was quite interesting. How we can do that? So the background is within this is the formula for the spatial resolution, and what you can see here is the temperature. Of course, when you're working with the biological systems, then this parameter is quite difficult to handle or to change, of course. Here are the parameters which are specific for the magnetic nanoparticles. So depends on the one side on the diameter of the particles, and also the saturation magnetization. And this the gradient strength. This is one of the parameters which is the hardware depending parameter, and which means by increasing the gradient, you can also increase the spatial resolution. And this is important. So that means For example, for the 330 micrometer, there was a specific system with 7 Tesla per meter. And so, yeah, increasing this parameter will also provide you better and better images, theoretically. But also the particle system is important. Here, for example, the same scanner parameters, but different particle systems. Here is a WIST, which is the contrast agent, the well-known contrast agent from the MR side, against specific for MPI developed tracer. And you can see, you can clearly distinguish between the small dots, just using an optimized particle system for that. Also, the real-time visualization, we spent a couple of years to optimize everything in the data processing. And here's a short movie. So on the right side, you see the real-time 3D reconstruction. And you also can hear with this beeps the imaging process, so the sequence running, and I just moved a point sample back and forth within the scanner, and it was one of the early tests for our reconstruction framework. And that brings us to the first in vivo images. And because of MPI just provides you the magnetic nanoparticle distribution, we have to get a background information, and we use this here from a biospec system, an R machine. And here on the left side, you can see the real-time in vivo experiment. And we administered magnetic nanoparticles on the tail vein of the small rodent, and you can see it goes up to the heart, and have a more specific look into this area. You can see directly the heart rate of the beating mouse heart. So that was one of the first time we can use, or we can show the in vivo experiment, and that everything works out within an MPI scanner. Since then, of course, you can do a lot of different stuff here. The intravenous administration of tracer, I showed you that, but we also performed experiments with in vitro labeled cells. So here is a small cancer lung model. So these cells are then go into the area of the lung. Or we also tried the in vivo labeling with functionless nanoparticles within graft versus host disease model, and everything works out quite well. We also tried functional MPI. So that means we put in a small rodent into the scanner and just the head in the field of view, because you know that the particle system is filtered quite rapidly from the liver. You can see that here. So after administration of the tracer, the signal in the liver starts to grow quite rapidly. And so we have to separate the areas. But then you can also see when you switch between carbogen and oxygen gas, you can see the difference in the brain signal. We also tried the combination with other technologies, methods. So the combination with MRI I showed you in the first slides, but we also tried out if it is possible to combine MPI technology within CT. For that, we built up a specific MPI scanner which has a CT window, and it was also possible to use both technologies simultaneously. And it was the first test for the next hybrid combination. So applications for MPI, we have a lot. Some of them I showed you, but there are also other groups working in this field and provides really, really impressive results with that. But one of our main focus is to use the speedness of this MPI machines, because as I mentioned that this is quite rapid and fast method. And this is, for example, for the interventional part in the medicine can be quite promising application. And we also tried this, and we do this for angioplasty, stenting, and instrument tracking. I will show you a bit in the next slides. But before I come to this, a short overview about different kinds of MPI scanners. So the TWMPI generation 1 I showed you, but there are also other ideas. So one of them, for example, on the left side is a field-free point scanner system, which was provided by by the Philips Group 2005. But there are also, from the Maxwell equations, you can use other shapes for encoding. And instead of a field-free point, you can also use a field-free line, which not only gets the signal from a point structure, more from a linear or from a linear structure. And so the advantage of this is from these FFL scanners are that they are more sensitive, but from the hardware side, it's more complicated and it's more sophisticated. You need more sophisticated hardware to run the systems. But in 2018, we found a way to overcome this problem. And so we came up with the next generation TWMPI scanner. And how does it work? I have a few animations for that. So let's start with two coils are assembled in a Maxwell configuration. And these 2 points, these two coils generates then a field-free point, or it's more an ellipsoidal structure. So this 3D representation here. And by adding a second coil pair, you can generate a field-free line. So standing or located in the center of these 4 coils. And now let's change the coils a little bit. So let's use this double cross coil system. And again, you can see here there's the linear structure. So it's still an FFL. And let's double this DCC coil system and put different currents on these 2 channels now. And let's do that in the same way as a traveling wave approach. So with the same frequency, the same current, but with different phases. And then you can generate not a traveling field-free point. In this case, it's a field-free line. And this is quite cool. So then it's quite easy from this point. Just add a solenoid. This solenoid now can be used for displacing the field-free line on a sinusoidal trajectory to cover a projection through the entire scanner. And for 3D, of course, in the same way as on CT is doing that, let's rotate the field-free line along the symmetry axis. And for that, we just have to assemble a second pair of DCC coil assembles. And with that, we are able not only to generate a traveling field-free line and a swiping field-free line, but also a rotating field-free line. And this is the first concept of a fully electrical driven FFL scanner. And this is really nice to have. And you can operate the system with 3 different frequencies. You can use different combination of frequencies, which is really interesting because this is one of the scanners which can be operated with different trajectories or different sequences. And you can imagine with the magnetization response of the different particles, you get also different influences of the sequences on the signal and on the images within such a scanner. And another advantage is these systems are really small. So you can see it here. This is one of the first prototypes, and a small animal scanner, so it has a bore size of about 40 millimeters, and it's really, really compact. And yeah, uh, one of my parts in the last couple of years is to bring up um, a commercial system, and we founded our company last year. It's Phase Vision, and we also provide these small systems, um, which are really, really nice to have. And I mentioned this, so the scan times is only 20 milliseconds per volume, and the systems are lightweight and easy to use. What you can do with that? So, for example, and this is one application we are also doing here at the university. We want to figure out what are the flow, for example, within an aneurysm system. And for that, of course, we have to extract first some real aneurysms from CT or MR data sets. And you have to prepare that in different steps for 3D printing. And then you have an elastic model of this really, really nice aneurysm phantom. And then you could put into this and compare the MPI data on the left side with different modalities. On the one side, for example, the optical transmission, or also as a gold standard, the 4D flow MR on the right side. And because the measurement time in MPI is really, really fast, you can, for example, here you can see that you can perform a lot of experiments per day. And for example, this one has been taken in 1 day. So the experiment can be done quite rapidly. But what we can do else? So we want to go to the clinic, and one possible application is the percutaneous transluminal angioplasty (PTA). And what is the background? So when you have an obstructed arteries, you can see it in the CT. So there's an absence of blood flow anymore. So we have some areas in your arteries which are which hampers the blood flow. And the treatment is quite interesting because you go in within guide wire, you put over the guide wire a balloon, and with this balloon we can open up this stenosis, and then hopefully afterwards everything is good again. And today this is done by continuous X-ray radiation, and you can see it here in a typical angio lab. You have to wear quite heavy safety equipment, and of course can be quite exhausting. And our goal was to get rid of this radiation technology and do that in the future with an MPI machine. So that was the idea in 2018, and we could show the entire procedure with our small preclinical system, and we do that within Phantom. So you could see the first to figure out, okay, we have here a stenosis within our aortic artery. Then you go in within guide wire, which is also visible in the MPI machine. Then you put over this balloon, and you open up this balloon with the MNP tracer, and afterwards you perform another angio to see, okay, there's a residual stenosis, so you have to apply a stent, and you go again with a balloon. The stent is also indicated by two areas, just to see them live in the system. And after after assembling the stand, everything is fine again. And this was the entire procedure just done by MPI. And our idea was, okay, so what we have to do to do that also on a human scale. And I learned you have to do a lot of stuff. So of course you have to keep in mind the sensitivity. You also need a high temporal resolution. You also need a good spatial resolution. And because the clinicians need that, you need a real-time visualization. And of course, the system should provide the in vivo capability. And so everything we did and we could show in the past. And the only thing we have to do now is to increase our systems. Just to see what's going on in the community. In 2016, for example, this was the first human-scaled MPI scanner provided by Philips, and it was a quite huge machine and really expensive. But yeah, Philips stopped this project, unfortunately. Then the UKE came up with a head scanner 2019, a really impressive system. And it's half a ton on a table and produces really, really nice images. Another big machine is provided at the MGH from Lawrence Wald. And it's a 2-ton rotating gantry for, with a bore size of about 30 centimeters for head scanning. Quite impressive machine. Also, Mitsubishi in Japan provides an system. And Magnetic Insight is a startup from Stephen Connolly in Berkeley, and they also built up a head scanner system. But all of them are not portable and are quite slow, so you cannot do real-time images for interventional purposes. So that means, okay, we have to think to maybe increase our systems. And just scaling up, to be naive in the first glance, it can be quite good. Just to go from 60 millimeters up to 1.8 meter, and just to make some calculations about that. So okay, let's build up a TWMPI scanner, which is large enough that you can put in a human, and you end up with some really, really high parameters. So currents about 1,700 amps, power dissipation of 2 megawatts, copper wire length about 700 meters, and this is only for one of the channels of one of the DLGA. So this is not possible. And that means, okay, let's think it a little bit different. Let's shrink down everything and just focus on maybe some extremities here, a leg. And within smaller gradients, which is high enough for good images, but also keeps the limit of SIR and PNS, which is also important to have in mind. And maybe a temporal resolution about 5 images, or here it's 5 seconds per image. It was the first idea, but at the moment we are at 5 images per second. And these parameters looks more usable. And we built up such a scanner with almost the same idea I showed you with a traveling wave field-free line approach. But it's a more specific system because this system not provides you 3D images. This system is optimized for providing 2D projection images, but with the advantage that you can use this system simultaneously with an X-ray system as well. Only 6 small steps to do. So starting with the simulation, CAD 3D printing of the parts. So that was quite nice work during the pandemic situation, and then the coil assembling, and then you can end up with a scanner. So this is how the scanner looks like. Here, my master student was the first volunteer, put in his leg into the scanner. and it's quite lightweight. So it's about 15 kilograms. It's portable. And step 5 puts some more parts on it. So again, the transmit receive train. You need a console with some peripheries. The amplification cabinet also. Again, these are common audio amplifiers. You need a real-time visualization part. And of course, you need some phantoms. So we built up a leg phantom with an here's also some artery structure out of the leg um, with an aneurysm, and for the more realistic data, a pumping system. You can see one of the first tests with the system. Again, all the software parts, the computer for sequences, here the cabinet, some filtering, and here the scanner. The first version of the scanner. Here's a sample filled with magnetic nanoparticles, and I put my arm into this and just run the system. You can hear it again, and with each beep, a full sequence is performed, and one image is taken. And on the right side, you can see in real time the reconstruction. This was the first test, in vivo test, so my arm is still working, of this scanner type. So with that, we can also provide quite good images, so regular vessels, also aneurysms or stenosis. We can also see the instruments, so here the guide wire and also the balloon catheter. And you can then perform an IMPI-guided PDA within a huge field of view. And again, the same steps, so the angiography, the balloon positioning, the balloon dilatation for open up the stenosis parts, and then the MPA after. And yeah, so everything worked out. And so this was done, or this was performed within our labs. And then we have the possibility to do that, to go into the clinical environment. And it was during the pandemic situation. And you can see here, this is the entire equipment you need for running the scanner. So everything is portable. You can just put some wheels under it, and then you can go into an angiographic catheter lab. So just run it once, and you can see it here directly into a realistic environment. And maybe again. So here's a bolus injected into the sample, into the leg sample within the scanner, and yeah, you get both informations at the same time, simultaneously, the X-ray and also the MPI information. So everything was cool. So what's about more realistic models? And in vivo, so far we are not allowed to do that with the living humans, but we had got the possibility to do that on human body donors. It was also quite new for me. So everything is almost the same. So you just replace the leg phantom by a real human phantom, and you put in on a pumping system, and then you can emulate the blood flow. So you can see it here. Here's the body donor. It's the same angio lab. So you got here the possibilities for the pumping, to add the pumping system. And then again, you can perform the same experiments. So putting in a bolus through the body donor, and you get also the IMPI results in combination with the X-ray results at the same time. You can track different boluses with different concentrations. Everything worked out for that. Really, really interesting. And we also get the possibilities to try out the first interpretations tests. So just to get an idea if there's some influences in image quality or is there some heating effects. And it turns out that MPI is really, really safe um, in this area. So there's no heating of protheses or implants, which is obviously also quite important. So what are the next steps before clinic? So we built up the system, done. We approved, we have an approved tracer. So since 2 years, we have the possibilities to get a Resotran, which is a copy, I would say, from Resolvis, but a better copy. We are at the moment we're working with the ethic committee the decision for the first in vivo images. We plan this. Then the next step, of course, the certification of our scanner system, and then in the future, the first cohort study and also the MDR approval. But maybe this is in 5 to 10 years. And just to see what we are doing at the moment. So this is the next version of our MPI, IMPI system. And at the moment, we are planning the first the first tests for PNS and SIR, and that would be the next steps within that. So I see it's more time than I thought I need. And there's another project which is also quite cool. This is the stroke project, which is a similar idea. So we are also using these magnetic nanoparticles for real-time tracking the perfusion in the brain. So at the moment we are at a good status. So we built up the first system and we could show that it is also doable in a realistic environment. Yeah, I want to thank my team, which was really, really great. And it helps me in every areas. And I also have to thank my partners and all the people which are supported us in the past, and of course you. And if you have some questions, then please let me know, and I'm happy to answer this.
Thank you so much, Dr. Vogel, for this really inspiring and interesting presentation. So I would like to ask if anyone have question for Dr. Vogel, can please go ahead.
Too late now.
Oh, no, maybe I can speak up. Sorry, Patrick, fantastic talk, but I just need to cough. Very impressive. Thank you so much.
Hi,
Amazing to
thank
see
you.
where, hi, yes, good to see you. Sorry, I was joining late. Impressive to see where MPI has gone and where your MPI has gone in particular. And I think it's impressive as well to see that this cross coil system allows you to really build lightweight systems. I mean, look at the tons of systems. I mean, metric tons in weight as they were before. I think this is really cool. My question is, the signal is really sparse, right? You have only usually very localized signals. And of course, if you think of line encoding, you sample some areas more often than others, and so on. And if you think of angiography, and I think your move is very smart to do this PTA comparison. However, still, as usual, we cannot beat the resolution of X-rays. So can you think of ways to improve that beyond the formula that you showed? Maybe reconstruction, maybe, I don't know what.
This is a good question, and we're also thinking about this topic in the last 10, 15 years, of course. And of course, one thing is the particle system itself. So we are here have a lot of groups thinking of other tracers and other tracer systems, and I want to highlight one of them. It has been provided, introduced 3 years ago. It's a super ferromagnetic material, and these particles have an quite impressive behavior because using these particle system within an MPI scanner can improve the spatial resolution by a factor of 10. And this was really impressive to see. Unfortunately, so far it's not possible to use these particles in an biological system, but maybe in the near future. From the other side, of course, the the scanning part, so the entire process, the data or acquisition process, it's one of the open questions also in my group, and one of my PhD students also working in this direction or looking in this direction to optimize the sequences and to get an idea about how we can improve the reconstructions and the reconstruction algorithms to get a better image quality, to make it faster, and also better spatial resolution. And of course, everybody is doing this at the moment. Also, artificial intelligence is one big question. How we can use this? Is this possible to also use this in this combination? And we also had some preliminary results. But I think in the next few years, a lot of things will happen, and one of them will be a better image quality, of course, and yeah, a lot of things to do, definitely.
Thank you. Before I was joining this meeting, I was at the hyperpolarization study group meeting of the ISPREM, and there we may face sometimes a similar problem as you, although I think you should be in a better position. You were saying that you're not allowed to do it humans. And maybe I really would like to talk to you more about that, because we're as well talking to our local FDAs and the federal FDA in Germany. But I mean, you're using Resovist, right? Which is or was an approved medication. And why can't you just A, do an individueller Heilversuch, or B, file a clinical trial?
Resovist is not anymore available, but the Bender group now provides this Resotran, which is similar contrast. So it's an approved constant contrast agent for MRI for liver imaging. It's also working quite well for MPI. And of course, you can use this in an off-label use for in vivo imaging. But at the moment, it is difficult to handle all the other open questions about safety, regulatories, and also the ethic committee and insurance thing and so on. But we are working on this, and I think we are much. So it's just a few weeks, month maybe, and then hopefully we will do that.
Maybe we can really talk more about this. I was really looking into the details of the laws, and there's several ways to do it. And this individual healing attempt, that's one way. Of course, the responsibility is always with you or with the doctor. Nobody takes away the responsibility, even if there's an ethical approval and so on.
Definitely, you're absolutely right. Yes.
But individual healing attempt, that's like my experience, the way you can do something tomorrow. Or what you can even today is, of course, the Selbstversuch.
Of course. We're also thinking about this. And let's say in this words, we
have one
in a planning phase. And so hopefully we will get this next milestones within the next time scale.
Yeah, it's very impressive. And if you don't manage it in Baden-Bayern or Baden-Württemberg, then you do it in Schleswig-Holstein, okay?
All right. Let's keep in touch with you. Of course. Thank you.
Okay. I'm not taking time away from other questionnaires. Thank you, Patrick. It was great.
You're welcome.
Thank you, Professor Jan, for your question. And now we have Dr. Mark, right?
Thank you very much for that talk. I enjoyed it very much.
Thank you.
I had just a couple of technical questions. At some point, I saw there was an amp at 1 kilohertz and another at 17 kilohertz, and maybe I wasn't paying close attention. Are you looking for intermodulation products?
Sorry. Can you repeat the last part?
Why the 1 kilohertz and 17 kilohertz? What was the point?
This is a good question. This is really a good question. So Philips started with an MPI scanner running or operating at 25 kilohertz, and I met Bernard Gleich, and he told me something like, so yeah, and it was 25 kilohertz because it's high enough that you cannot hear it anymore, because it's quite annoying when you have this sound around you. And on the other side, you can use common audio amplifiers. But on the other side, it turns out over the time that going to higher frequencies, you have the problem that then the Brown relaxation effect comes more into play, and you get a problem of the more blurring of your images. And I think it was about 10 years ago. So there's a group in Turkey, and the group showed that when you go down to quite low frequencies, then you can overcome this Brownian relaxation effect, and the spatial resolution of your images are get better and better. On the other side, of course, the disadvantage of lower frequencies is you have a lower induction signal into your receive chain. So this is at the moment a little bit the difficult, or there are different ways to overcome this as well. And on the other side, you also have to keep in mind the PNS, because lower frequencies, for example, the Lex scanner uses 60 hertz for the main field and 2.5 kilohertz for the swiping field. And in combination with a gradient of 0.3 Tesla per meter, which is quite low compared to a preclinical system, where the gradient is in the range of 2 to 3 Tesla per meter. But the fields, the magnetic fields you need in combination with the frequency, you get quite near to the limits of the PNS you can use for humans. And so you have to go down with the frequency. You have to change a little bit the idea from the signal generation within an MPI, because you are limited from the regulatory side. And for preclinical systems, everything is fine. But thinking in human scale, you have to go down with the frequencies. We also think to go further down, just to also to have the possibility to increase the gradient again to get better spatial resolution. But then you also have to optimize the receive chain for better sensitivity and so on and so on. So it's difficult to handle. So a lot of open questions and a lot of different ways, and so the community has different ideas. So it's also quite interesting to see, and we will see what's coming up. Yeah. But this is the main point. So going to higher frequencies with a classical encoding scheme is not possible. So you have to go down to make this encoding in the classical way. When you want to use higher frequencies, then you can do a so-called single harmonic acquisition. So that means that you have a quite slow gradient system just for moving a field-free point slowly through the field of view, and you're using a high frequency field with a low amplitude, just to produce the second harmonic of your particle system, and to get the signal in that way as well. So this is also possible, and there are also scanners in some groups working exactly with this idea. But the disadvantage is that the systems are much slower. So you need a couple of minutes for one of the slices. So depends what you want to do.
Thank you.
You're welcome.
Okay, thank you, Mark, for your question. Do we have other questions?
No more questions?
No more questions. So thank you again, Dr. Vogel, for your time and your inspiring presentation again.
Yeah, thank you for the invitation.
Yes, thank you for all participants who came today, and see you next time.
All right. Yeah. Thank you
Thank
so
you.
much. And bye-bye.
Bye-bye.